Okay, great. So my name is Niall. I'm going to be discussing some of the work I've done throughout my PhD. I'm going to ask the question here about the importance of focal lesions and their relation to what are classically described as being frontal behaviours. A lot of the work that I'm going to be discussing is going to be in relation to social cognition, which has been a theme throughout my PhD. So to begin with, instead of giving an overview of the different aspects of this presentation, I'm going to ask some very particular questions. And these are going to be focused around these three things. So the first, what are the neural correlates of frontal behaviors? So trying to address the question of linking particular behaviors such as apathy, disinhibition to frontal lobes. Two, looking at holistic approaches. So not focusing on what is obviously bad. So in many cases, after a traumatic brain injury, there may be some focal damage. And it's very easy to try and focus on the site of these focal injuries and relating that to the behavioral impairments that are seen. But due to varying mechanisms of damage, such as the acceleration-deceleration forces following road traffic accidents, there may be more widespread damage rather than just the focal injuries that might occur from penetrating injuries. And three, there's various different things that we can look at or various old questions in relation to the brain behavior link with novel tools that are constantly being developed. We always need to look at these new methods and seeing if we can try and address some old questions by looking at these problems in new ways. And as I mentioned, I'm going to do this in the context of social cognition following traumatic brain injury. So this is simply just a PubMed search for the terms TBI and social cognition. We've got quite used to looking at these types of exponential curves recently. So there has been an increase in the publications of TBI and social cognition. I should point out that the vast majority of these are in paediatric samples and not adult cohorts. But I would draw attention to the y-axis. So last year in 2019, there was close to 40 publications with these terms. And this is quite a small number in comparison to other psychiatric and neurological conditions such as schizophrenia or Parkinson's disease, where much more work is being done on social cognitive impairment. So this is an area that has been under-researched following traumatic brain injury. So what is social cognition? So this broadly relates to the functions and behaviors that allow us to integrate and social behavior. And one of the core paradigms that are frequently used are emotional recognition paradigms. So trying to probe if somebody can recognize particular emotional expressions. This has been quite frequently done through the Ekman Faces, which is a standard stimuli set. For a lot of my work, I use the Chicago Face database within this new platform developed by Adam Hampshire's team, with a lot of the heavy lifting in terms of coding the paradigms done by Will Trender. This is quite a nice platform as it allows quite dynamic implementation of tasks through either smartphone, tablets, or on the web. And what it would allow us to do is run the same paradigm, but with different stimuli sets. So we could try new stimuli sets and seeing if the same emotional recognition deficits or abilities are seen with new stimuli sets and if they generalize. And it makes me quite happy to know that social cognition is a big area of research within canines. There's even some eye tracking paradigms that have been conducted. So why is it important that we look at these expressions? Well, for one, it helps us to identify potential threats if we are seeing angry expressions, but also just in normal interactions. The feedback we get from emotional expressions helps with social bonding. We need to know how somebody might be thinking or how they might feel in terms of in particular situations. And to kind of be able to process that feedback. Just to point out here is quite a cool study, which I liked. So you might be familiar with the monkey ladder test, which shows chimpanzees have this incredible working memory capacity. While dogs don't have the same working memory capacity, they do have this exceptional ability for recognizing human emotions. This makes sense in terms of they've been domesticated, and their ability to understand human expressions is quite important to their cohabitation with us. And this is in contrast to wolves, which don't have this shared ability. Now, of course, as well as being able to test these behaviors, we're also quite interested in what's going on in relation to neural functioning. So there's various tools that we have to investigate this, particularly with MRI. I want to point out the importance of looking at social cognition, particularly following traumatic brain injury. So there may be quite dramatic impairments or problems relating to quality of life due to breakdowns in interpersonal relationships as a result of impairments to social cognitive processes. So I'm just going to run through this conceptual model of social cognition. So at any given point, an individual might have a particular state. They might be feeling a bit anxious, they might be feeling a bit low, and they might have a bit of fatigue. Of course, there may be positive aspects to an individual state as well, but this state influences various different aspects of how we perceive information. So while we're interacting with another person, there's various steps that may occur. So this is a perception. How do we recognize the expression of the other person? Do we understand the context in which we're interacting with this other person? So from kind of psychiatric literature, we know that depression may negatively influence how we perceive emotional expressions, creating this negative bias. So this leads to how do we process the information? So how empathetic are we towards another individual when we're receiving information from them? How do we judge a particular situation, and how do we incorporate the information from a situation with our prior knowledge and experience? And how does this then inform how we interact or influence our decisions that we make? Now, none of this happens in isolation. This is a very dynamic process. And while this is occurring, we're constantly receiving this feedback. And there's this idea of metacognition, which is primarily being looked at in terms of education. But it's quite important here in social cognition as well. So we're constantly evaluating our behaviors, and this is important. So metacognition is thinking about thinking. So are we monitoring our own behavior to see if we're acting appropriately given a particular situation? And this is quite relevant to traumatic brain injury. So anyone who's picked up an introduction to psychology or neuroscience textbook will be familiar with the case of Phineas Gage, a 25-year-old male with a penetrating injury through the frontal lobe while working on the railroads. And it was reported after this injury that Phineas had very dysexecutive behaviors, had dramatic personality changes. And this has led to one of the quintessential case studies of neuropsychology linking brain location and behavior. And it's been associated, frontal lobe damage has been associated with a variety of symptoms, including dysexecutive behaviors, apathy, and disturbance to mood and personality. And this can have very dramatic implications in terms of how one engages in rehabilitation, their interpersonal relationships, and getting back to work. There's various risk factors that may be associated as well, including increased social isolation due to inappropriate behaviors with others. It's also known from the prison population that there's a high number of inmates with traumatic brain injury, and there's an increased risk of assaults also. And this may arise due to inappropriate judging of others' intent. So there's various tools that we have available to us. So I've mostly focused with MRI, in particular looking at diffusion measures, as well as looking at white matter and grey matter volume, and looking at dopamine transporter uptake with SPECT imaging. There's also resting and task-related fMRI that we have available to us as well. So I'm just going to highlight some important regions, but it's important to note that there has been a shift away from localization of function to network theories. So we're not interested in isolated regions, but more so how these regions might be structurally, functionally, or chemically connected to each other. So we have the medial prefrontal cortex, which is important for executive functions, the anterior cingulate cortex, which is being highlighted in a wide variety of functions, including decision-making and error monitoring. The striatum, which has very high density of dopamine transporters and is important for executive functioning. So we can look at the structural connections as well as the dopaminergic projections from a variety of different regions. And we also need to consider how these might be functionally connected also. So there's a variety of different neuroimaging measures that we can use. So we can look at the diffusion measures such as fractional anisotropy, white matter volume, the functional activation, as well as looking at dopamine transporter uptakes. So just going back to this case study of Phineas Gage, this was an N of 1 and provided us with some useful insights into how the brain and behavior might be integrated. But we can develop a little bit on this. So what I've done is taken 102 moderate severe traumatic brain injury patients and looked at the lesion distribution. So here we can see that the burden of lesions primarily falls within the frontal and temporal poles. And now that we've got increasing samples, it allows us to move on and do some more sophisticated analysis in terms of linking to behavior. So I've used this voxel-based lesion symptom mapping approach, which has been quite widely used within the stroke literature, and to a lesser extent within traumatic brain injury patients. The first thing I want to focus on is looking at this measure of frontal systems behavior. So this is a neuropsychological assessment looking at behaviors that are classically attributed to being relevant to the frontal lobes. However, there's been quite little done in terms of actually looking at the neural correlates to this measure. And we can see here that with increasing symptom severity of the frontal systems behavior rating scale, you have a higher chance, there's a higher link to having lesions in the right inferior frontal gyrus and temporal poles. When looking at other neuropsychological measures, such as the Stroop inhibition, we see more bilateral links to frontal lobe lesions, including the temporal poles. And with the switch cost from the trail making task, the association with lesions is more associated with the medial and orbital frontal cortex. So one of the questions I wanted to address was, what about the moderate severe patients that are classified on the Mayo rating scale that don't have focal lesions? So they might have been classified as being moderate severe due to duration of post-traumatic amnesia, duration of loss of consciousness, etc. And looking across a range of cognitive tasks, including the FiTBI, processing speed and executive functioning, what we see is that there is no significant behavioral difference between the lesion and non-lesion moderate severe TBI patients. And this is an important finding. So it really highlights that the absence of focal lesions does not indicate that there won't be a presence of severe behavioral impairment. So we can move on to more advanced imaging measures such as diffusion tensor imaging. And just to give a quick short primer, if we were to drop ink into a glass of water, this ink would disperse randomly throughout the glass, and this is known as Brownian motion. However, if this ink was dropped within a straw, it's now constrained by the axis of this straw in what directions it can travel. And this is the main principle of diffusion tensor imaging. So we've got various different tissues within the brain with various different diffusion properties. So in the cerebral spinal fluid, due to this random motion, we have quite quick signal drop-off. Whereas in the white matter, such as corpus callosum here, we have hindered or restricted diffusion along the directionality of the axons. And this gives us a signal which we can measure. And we use this as a surrogate measure of the structural integrity of the white matter. And this is the principle of the diffusion tensor imaging model, where we have a primary eigenvector, which allows us to measure the directionality. And we can see here along the corpus callosum in red, this is a primary left-right directionality. In blue, these are the ascending, descending fibers, cortical spinal tract. And in green, these are the anterior, posterior fibers. So when we compare, when we look at our TBI patients compared to controls, on the top row here, we have our lesion patients, and with the effect of lesions regressed out on an individual basis. And we can see that even with the effects of the lesions regressed out, we have quite widespread white matter damage. But interestingly, in our non-lesion TBI patients, again, in red here, we see quite widespread damage. And this kind of comes back to this idea that there might be processes such as the acceleration-deacceleration forces from road traffic accidents that lead to widespread damage in the absence of focal penetrating injuries. But importantly here, when we can contrast our lesion with non-lesion moderate severe TBI patients, we see very little difference. So there is some difference here within the genu of the corpus callosum. On the right, I've plotted the effect size differences of the lesion and non-lesion groups compared to healthy controls. And we can see between these groups, there's no significant difference apart from the genu of the corpus callosum. Interestingly here, our non-lesion TBI patients have a significant difference compared to controls within the cortical spinal tract, whereas this effect is actually not seen in the lesion group. So this might indicate some varied pathological process which has occurred compared to the type of injuries which may lead to focal damage. So I just want to highlight this idea of the disconnectome. So this is work that's been done by Michel de Schotten, and has primarily looked at this in terms of stroke literature, but I think it's quite an interesting approach and is kind of an area that's being developed. So I've done some preliminary analysis, and we're quite happy to have discussions about potential ways to look at this going forward. One of the things I did was constrict the lesion probability distribution to the highest probable region, so the top 10%, and use this as a seed region for probabilistic tractography to identify the white matter tracts which are most probably disconnected due to presence of a focal lesion. And we can see these here. But I also extended this to look again with this lesion mask here. Using masked independent component analysis, I applied this to 100 healthy controls from the Human Connectome project to see if the region that's most probably lesioned, if that can be functionally disassociated into different functional networks to try and identify if kind of various functional networks might be impaired depending on the site of the focal lesion. As I mentioned, this is kind of developing work, and be interested to hear people's thoughts or discussions on how this might be brought forward. So going back to some of the diffusion work, there's new methods which allow us to acquire multiple diffusion weighted images at the same time. And this multishell approach allows us to look at more sophisticated models, such as the neurite orientation and dispersion indexing model. This is proposed to allow a more biologically meaningful estimate of various aspects of white matter structure that might be affected due to differing pathological process. So we have the sticks here as part of the neurite density index, which is suggested to be more related to the neurites, so axons and dendrites, whereas the orientation dispersion is more related to the overall structure of the tracks. And we also have the cerebral spinal fluid here measured as a separate compartment, so potentially removing noise from the other estimates within the model. So here we've seen quite interesting spatial variation in terms of impairments that are seen. So one of the important things to highlight here is that volume in blue actually has quite a large effect size across all the different tracks, even in regions where there's no significant effects picked up within the diffusion measures. So FA in red can be partially broken down by the neurite density in orange and the orientation dispersion in green. And these, as I mentioned, these show varying spatial patterns. However, while the volume has the greatest effect in terms of identifying group difference, it seems to be very non-specific in terms of relating to cognitive functioning, where we see various relationships within the diffusion measures. The particularly interesting ones I want to highlight here are to do with the choice reaction time, or CRT, associated with a negative main effect of neurite density index in quite widespread regions, whereas when looking at it in terms of mean diffusivity, it's constrained to the cortical spinal tracts, which I think is quite interesting because the mean diffusivity might be constrained to here with a relationship to what might be a primary slowing due to motor reasons, whereas with the neurite density, it might be associated with a more widespread cognitive slowing. I do need to highlight here that there's quite a high overlap with these measures, and they're not completely independent. So I just want to come back to these three things. So what are the neural correlates of frontal behaviors? I think it's important to really stress the point that there are regions that might be preferentially involved in particular behaviors, but these are more likely to be related to a network that is important for these particular behaviors rather than particular regions in and of themselves. So it's important to look at holistic approaches and not just focus on one particular aspect of trying to address a question. And as I showed with the disconnectome slide, there's always new developing tools and ways of looking at old questions, and these are important for helping us understand the brain behavior relationship to a better degree. And I'd like to thank everyone in my lab and everyone that's been involved, including collaborators throughout my PhD, and have contributed to this work. Thank you very much.